Good afternoon, everyone. My name is Jun Yin Lum. I'm a third-year entomology major graduating this quarter. My faculty mentor is Dr. Chaoyang Li, and today I'll be talking about the anthropogenic influence on long-legged ant and Argentine ant distribution. So to start us off, I'd like to introduce the two species that I focused on. On the left, we have the long-legged ant, Anopolepis chrysolipes. On the right, we have the Argentine ant, Linepitima humili. And what both of these species have in common is that they are widely considered to be major tramp ants and among the top 10 worst invasive species in the world, as ranked by the Invasive Species Specialist Group. So what are tramp ants? Well, the definition varies depending on who you ask, but in general, they are ants which are strongly anthropophilic and reliant on humans for long-distance dispersal. Because of their close association with humans, they have invaded many parts of the world by hitching a ride along trade routes. And in places where they have been introduced, they have become major ecological, agricultural, and household pests. These are some of the impacts of the long-legged ant and Argentine ant. In areas where they have been introduced, they reduce native ant diversity by outcompeting native ants. And in severe cases, they can even lead to declines in populations of other arthropods and wildlife. In agricultural settings, they protect sap-feeding insects like aphids, resulting in outbreaks. And they are also a nuisance in and around human dwellings. The long-legged ant in particular is most infamous for causing an ecological meltdown on Christmas Island, where they killed large numbers of the native red crabs and led to large-scale changes in the forest structure. As mentioned before, both of these species of ants are tramp ants and they are associated with human activity. However, limited research has been done on the effect of different levels of human activity. Previous studies on the Argentine ant in San Francisco and the long-legged ant in Taiwan found that both species are most abundant in areas with moderate human disturbance. This leads to the main question of my study, which is how different levels of human activity affect the distribution of long-legged ant in my hometown of Penang and the Argentine ant in Riverside. I hypothesized that the highest ant abundance will be observed in areas with moderate or high human activity. For each species, I selected 3 sites with a gradient of human activity. Each site will have 4 sublocations corresponding to levels 1 to 4 of human activity: low, moderate, high, and very high. To measure human activity, I counted the number of passers-by over 30 minutes and used the average number of passers-by to classify each sublocation according to the 4 levels of human activity. To measure ant abundance, I placed 10 baited index cards with honey and peanut butter around each sublocation and took photos of the cards after 30 minutes. In addition, I also took temperature and humidity recordings for each sublocation. All the surveys were carried out between 8 to 10 a.m. and repeated for 6 or 7 days. This image shows the study sites in Penang Island, located in Malaysia. So the 3 study sites are Youth Park, Bukit Jambu, Relau, and Sungai Ara. These are the study sites for the Argentine ant around UCR. These are the images showing the baited index cards and pictures of me doing the surveys. The picture on the top left shows the long-legged ants, and the picture on the bottom shows the Argentine ants. After the surveys were complete, I scored the ant abundance on each index card according to the following categories. After that, the abundance scores were pulled and averaged for each sublocation according to the sampling date. To analyze my data, I used generalized linear modeling with Poisson regression. The dependent variable was the average ant abundance score, while the independent variables were the natural log of the numbers of passerby plus 1 and its quadratic term, along with temperature and relative humidity. I also conducted further analysis with non-parametric one-way Kruskal-Wallis test followed by pairwise Wilcoxon rank sum test to determine if N abundance significantly differs across the 4 human activity levels. All the analyses were performed with JMP version 15. These are the results from the surveys of the long-legged ant. As you can see, in the areas with low human activity, with the exception of the Bukit Jambu and Rilau site, the other 2 sites showed a relatively low abundance of the long-legged ant. In the areas with moderate human activity, the ant abundance was relatively high, with the exception of the youth park site. And as for the locations with high human activity, the long-legged ant was moderately common across all the 3 areas. And lastly, for the areas with very high human activity, the long-legged ants were almost absent. So these results are also reflected in my analysis after using the generalized linear modeling, where I found the number of passersby to be a significant predictor in influencing the average abundance score of long-legged ant. The black dots show the data points from my surveys, and the blue line is the regression line that was generated from the most parsimonious model. There is clearly a peak where the number of passersby corresponds to moderate human activity, as shown here. For my further analysis with the non-parametric test, this figure shows the results. And the different letters show the columns with different letters represent significant differences. So we found that only the N abundance was significantly lower only in the areas with very high human activity, but in areas of moderate human activity, the N abundance was higher, but not significantly higher than the other 2 levels. In contrast, for the Argentine ant, I did not observe any obvious trends in terms of human activity and abundance, although there was one location with high human activity where the ants were extremely abundant. After I analyzed the data with the generalized linear modeling, I found that none of the factors that I measured were actually had any significant effects on the abundance of the Argentine ant. As you can see, the ant abundance did not vary across different levels of human activity. So for the long-legged ant, the results show that its distribution was significantly correlated with human activity. There could be a number of reasons for this. Firstly, human-modified habitats may have more suitable microclimates as a result of reduced vegetation cover and shade. Secondly, human-modified habitats could have increased propagule pressure as a result of human activity, bringing in new introductions of the ants. Thirdly, areas with human activity may have increased resource availability in the form of honeydew-producing insects and nectar from irrigation, or it could be in the form of human foods, which the ants can feed on. However, the ant abundance was lowest in areas with the highest levels of human activity, which could be due to increased interspecific competition with other tramp ants and reduced nesting site availability. During my surveys in Penang, apart from the long-legged ant, I also collected 5 other species of major tramp ants. These are the tropical fire ant, longhorn crazy ant, ghost ant, destroyer ant, and robust crazy ant. And all these 5 species could potentially compete strongly with the long-legged ant. This graph shows the average number of major tramp species and the percentage occurrence of the long-legged ant across the 4 different levels of human activity. So as you can see, there is a general trend of the tramp ants, the other major tramp ants being more commonly found in areas with high and very high human activity. And this is where the long-legged ant becomes less common as well. In terms of the nesting sites for the long-legged ant, they most often nest in rocky crevices under boulders and foundations. They also commonly nest under fallen vegetation and other miscellaneous debris, but rarely construct nests in soil. Areas with very high human activity have a higher proportion of concrete structures, and these areas are also usually more well-maintained, resulting in a general lack of debris and a reduction of suitable crevices in man-made structures. Therefore, areas with very high levels of human activity could have fewer suitable nesting sites for the long-legged ant. As for the Argentine ant, as mentioned previously, its abundance was surprisingly not affected by any of the variables measured. One possible explanation is that its abundance could still potentially be affected by other variables which were not assessed in my survey, such as soil moisture. Alternatively, the lack of observable trends could be due to the small geographical area surveyed. However, similar to the long-legged ant, interspecific competition with other non-native ant species could affect the abundance of the Argentine ant as well. During my surveys at UCR, I encountered the two other pest species of ants, the red imported fire ant and the dark rover ant. And again, when the number of other pest ant species was higher, the occurrence of the Argentine ant tends to be lower, although it is unexpected that these other pest species also tend to be encountered in areas of moderate human activity. In conclusion, the results for the long-legged ant matched my hypothesis. The highest ant abundance was observed in moderate human activity areas due to suitable microclimatic conditions, high propagule pressure, increased resource availability, reduced interspecific competition, and abundant nesting sites. On the other hand, the results for the Argentine ant did not match my hypothesis, as ant abundance was not affected by human activity, but more research is needed to investigate this. Still, Argentine ant distribution may be affected by the presence of competing species. Thank you for listening to my presentation. I would like to take this opportunity to thank my faculty mentor, Dr. Chaoyang Lee, for his constant support and guidance, which helped me complete this project, which was partially undertaken remotely. His mentorship has been truly invaluable towards my personal growth and development as a researcher. Next, I would like to thank Sue and Dr. Chu for their assistance with the statistical analysis and modeling, as well as Greg, our SRA at the Lee Lab, for teaching me how to capture high-quality images on a lab microscope. Additionally, I would like to thank my parents for always supporting me in my endeavors, and especially my mother for driving and accompanying me to the various study locations. Lastly, I would like to acknowledge the funding that was provided by the UCR Undergraduate Mini Grant Program and the Carlstrom Western Exterminator Company Scholarship. These are my references, and I'll gladly take any questions.
Thank you.